So what we're going to do today is we're going to start a whole new topic. We're going to start into the cardiovascular system. And we'll begin here with thinking about the mammalian heart, its structure and function. Basically, we're going to walk our way through the chambers, which are the rooms of the heart, the tubes leading in and out of the heart, blood flow through the system, and then finally, we'll talk about the valves separating the chambers, and the valves that then are effectively the doors that are in the rooms and the doors that allow exit from the rooms, and we'll walk our way through that. As we begin to talk about this, the first place I want to start is really at basic principles of why even bother to have a heart. And we need a heart because we are three-dimensional organisms. We are multicellular organisms. In the old days, when we were single-celled organisms, we really didn't need to have a pumping system because the plasma membrane is all that separated the cytoplasm from the outside of the cell. Then once these creatures began to evolve into multicellular organisms, the first round of multicellular organisms were things like the jellyfish, the cnidaria. And the jellyfish are basically two cell layers. They are an outside cell layer, an inside cell layer, and the pumping system in the jellyfish is to simply move seawater between the two over the cell layers and use the simple diffusion from the water into those cells. So even in those animals, there's not really much need for a proper pump to move blood to within a couple of millimeters of all the cells. What you have to recognize is that we come up into the limits of diffusion pretty rapidly. Once we get more than 1 or 2 millimeters away from the source of oxygen and the source of nutrients, then the diffusion paths become too long to sustain life in those cells. So effectively, what we do with a heart is we develop a pump. The heart is a pump that brings the blood to all the cells. Effectively, we are bringing the ocean, our own internal ocean, to all of the cells in our body. And we're doing that by pumping the blood through the cardiovascular system. Now, I want to introduce the idea of the heart by simply talking about a fish heart. And we're going to talk about a fish heart as having only 2 chambers. The upshot of this is going to be that in a fish, you have one pump. A pump that is made up of an atrium and a ventricle. The atrium receives the blood, the ventricle then is the pump which moves the blood. But this single heart moves blood first to the oxygenating organ, which is in a fish, a gill, and then the blood moving through the gill is then distributed to the rest of the systemic vasculature. One pump, both for the oxygenating organ and for the rest of the vasculature. That's the case of the fish heart. But once we begin to look at particularly homeothermic animals, these warm-blooded creatures, which include the birds and mammals, then it appears the pump needs to become more complicated. It may be that the pump that we see in birds and the pump that we see in mammals have followed parallel routes to form a 4-chambered heart. But regardless, we have 4-chambered hearts in mammals and birds. The 4 chambers really constitute 2 pumps organized in series. So where the fish heart was a single pump that had to pump blood throughout the entire system, once we begin to look at 4-chambered hearts, we now begin to see 2 pumps. We see a pump, which we're going to call the right pump. This is the pulmonary pump. It's dedicated to pumping blood to the oxygenating organ, which in birds and mammals, of course, will be the lung. Then the blood which is pumped through the pulmonary circuit to the lung in birds and mammals, that pump now is going to be pumping the blood through the pulmonary capillary beds and returning that blood back to the left pump. The left pump is going to be dedicated now to pumping blood throughout the rest of the circuit. So all of the blood that goes to your brain and your liver and your gut, your muscles, all of that is pumped by the left pump. But first we use the right pump to oxygenate the blood, and then we use the left pump to pump that oxygen blood throughout the system. But when we look at the absolute structure of the left and right pumps, the left and right hearts, we find they have a very similar organization. There's a right atrium, and a left atrium. And in both cases, the atrium is receiving blood. The right atrium is going to be receiving deoxygenated blood from the vena cava. The left atrium is going to be receiving oxygenated blood from the pulmonary vein. The ventricles then become the major pumping structures. The right ventricle is going to be a chamber, but that chamber is going to be surrounded by a wall of muscle. That wall of muscle, called the myocardium, when it contracts, when the myocardium contracts, it's going to force blood out of the chamber, the ventricle, out into the pulmonary artery and carry blood away from the heart to the lungs. Similarly, on the left side, oxygenated blood will enter the atrium, pass across a valve between the atrium and the ventricle, and then when the ventricular myocardium contracts and generates pressure on the fluid within the chamber, as that pressure builds, it's now going to open the aortic valve, and that blood now is going to be pumped out into the systemic vasculature. We're going to walk through the movement of the blood through these different chambers. In the first iteration of this, I'm going to simplify it a whole lot. I'm not going to put any muscles in. I'm just going to make 4 rooms, 4 chambers. And then I'm going to put valves I'm going to put a series of valves which will constitute doors between the chambers. And we'll then lay out the valves and the valve structure in this 4-box model. In the 4-box model, we're going to find that there is a left and right atrioventricular valve. There's a valve between the atrial chamber and the ventricular chamber, and we'll call these atrioventricular valves. There's some other more arcane names we'll walk through, but if you hold on to atrioventricular valves, left and right, that'll be fine. And then there's going to be a pulmonary valve. The pulmonary valve lies as a door. It functions as a door between the chamber on the right side and the tube called the pulmonary artery. Then there's an aortic valve on the left side. On the left side, the aortic valve serves as a door between the left ventricular chamber and the tube leading out of the left ventricular chamber called the aorta. That will then bring us around to this nomenclature, and I'll try to bring it up early. The nomenclature that you need to hold on to, and the ones that will serve you well when you're thinking about the kind of blood flowing toward and away from the heart, is the convention. Anytime we talk about an artery is carrying blood away from the heart. Anytime we talk about a vein is carrying blood toward the heart. If you hold on to that nomenclature, it'll make things a lot easier as we work our way through this. So let's begin here with a simple idea of why have a heart. And it has nothing to do with the Tin Man. There was a time when we were single-celled organisms. And under those circumstances, this was basically the ocean. And this was cytoplasm. And as we have seen, the only barrier between the ocean outside and the cytoplasm was the plasma membrane. Under those circumstances, diffusion can do most of the work for you. We can have an oxygen gradient. So there's more oxygen in the ocean than in the cytoplasm, so oxygen diffuses in. The byproduct of oxidative phosphorylation in mitochondria, the byproduct being carbon dioxide, now can simply diffuse out. It easily diffuses across the phospholipid bilayer. Similarly, the byproduct of breakdown of amino acids, which is going to be ammonia. Ammonia, again, is water soluble and easily diffuses out into the extracellular space. When we start talking about kidneys, we'll recognize that ammonia itself is pretty toxic. And so as mammals, we're going to convert that ammonia to urea and get rid of it in that way. But still, ammonia is a byproduct of cellular metabolism, particularly of breaking down proteins. And then finally, of course, there will be nutrients. Out in the ocean, and those nutrients in many cases can simply diffuse across the phospholipid bilayer and enter the cell. So fundamentally, when all that separated the ocean from the cytoplasm was the phospholipid bilayer, we could simply depend upon simple diffusion. That case even pertains when we talk about things like jellyfish. Talk about jellyfish and we talk about sponges. These creatures are organized pretty much as having only a couple of cell layers. There's going to be an outer cell layer, and an inner cell layer. And then the way the animal's organized is that water moves through this open cavity and out to the outside world. And since the fluid is moving by a single layer of cells, the outside is facing the ocean. And we simply move fluid through this tube, and again, we can use diffusion to deal with oxygen, carbon dioxide, and ammonia. The problem arises in dealing with these various molecules that can only enter cells by diffusion. The problem arises when we begin to find three-dimensional organisms. Organisms commonly referred to as metazoans. Look around us. I mean, most of the organisms we look at, whether it's flies or humans or cats and dogs, those are three-dimensional organisms. Once you generate a three-dimensional organism, then a whole new set of problems arise for this diffusion. So, what I'm going to call metazoans, beyond the jellyfish and the sponges, require a pump to move blood, which in a way is an internal ocean, to within 1 to 2 millimeters of all cells. So once a creature expands beyond these limited diffusion distances of no more than a millimeter or so, then we have to invoke a means of moving fluid containing oxygen and nutrients to those cells, and also use that fluid then to sweep away the carbon dioxide and the ammonia, which are the waste products. So that's the reason for the pump. And so if we think about this, and we start with something as simple as the fish circulatory system. Circulatory system of a fish. The story I'm going to tell you would be pretty much the same even for the jawless fishes like those primitive hagfishes, but we're going to draw something which looks more like a fish, or at least for me looks more like a fish, although these will be bad drawings. There's the dorsal fin. Here's the head. There's an eye. And there's a mouth. And in orange back here is something known as the operculum. The operculum is the structure we look at on a fish, on a teleost fish, and we see it moving in and out, and we call that the gill. But that's not truly the gill. This is really a kind of bony structure. And what it is a kind of bony flap that extends over a very, very delicate series of structures beneath it called the gill. And if we look at a gill, the gill is an oxygenating organ. It's pretty simple, however. So what I want to draw here, this finger-like structure, is meant to represent an epithelium. So each one of these hash marks is meant to represent an epithelial cell. So this is meant to be the gill epithelium. And then immediately beneath this is going to be this tube. And this is going to be a blood capillary. Now, I'll try to draw this in and recognize now that there's a single cell layer of a capillary endothelium. Since we've split open this structure, we're going to see an endothelium on both sides. But as the blood flows through here, what I want you to recognize is that we're only two cell layers. The inside of this tube, where we have blood, inside of the tube where we have the blood is only separated from the outside world by the endothelial cell layer and the epithelial cell layer. So we can now imagine a situation where oxygen can easily diffuse across the epithelium, across the endothelium, and enter the blood vascular compartment. Similarly, we can imagine CO2 diffusing in the other direction. So effectively, we have solved this problem of going all the way back to a single-celled organism by bringing the blood right next door to another epithelial cell layer on the outside and allowing diffusion between the two. But if we ask the question, how do we move blood through that system, then we have to describe the fish heart. The fish heart is composed of an atrium, and connected with that a much thicker walled structure called a ventricle. Coming out of the ventricle is a single tube that carries blood to the gills. The atrium, I'll put this in blue, the atrium is collecting blood. This is deoxygenated blood. So the deoxygenated blood is flowing into the atrium, is being pumped then by the ventricle through this tube to the capillaries in the gill. And at this point now, we can oxygenate the blood, and oxygen-rich blood leaves the gills. This goes to all systemic organs. So the blood that comes through the gills and is oxygenated in the gills, that blood now is distributed to the brain, the liver, the muscle, throughout the animal structure. Well, this really is not an especially efficient system. Far be it for me to criticize fish. They've done just fine for several hundred million years. But the system is set up in such a way that all of the force that's generated on the blood here in the ventricle, much of that force is dissipated when the blood moves through the fine capillary beds of the lung. Then a lot of that energy simply has been absorbed, but the low flow, the low pressure output goes to the rest of the vasculature. One would think that if you were going to design this thing from first principles, that you would put another pump on the far side of the gills. In other words, you would use this ventricle to pump blood through the gills, then use another pump on the far side of the gills to move the blood to the rest of the system. But fish never did that. Oddly enough, squid did. So this is an aside here. If you look at squid, and they're highly metabolic invertebrates, squid have figured out a way of generating a pump that is a gill pump, and then on the far side of the gill, that blood is picked up by another pump, and that blood is pumped out systemically to the rest of the vasculature. For cold-blooded creatures living in the ocean or living in fresh water, this seems to work fine. But once animals started to climb out on land, and once they started to breathe not with gills, but with lungs, everything changed. And the culmination of that change is going to be the four-chambered heart that we see in mammals and birds. So in this upper panel, I'm going to take this panel and describe the four-chambered heart in a kind of four-box model. The emphasis in this story is going to be in the chambers. We're just going to follow the movement of blood through the rooms, as it were. And then once we've laid out the geography of the rooms, once we have sort of a floor plan for the heart, then we'll start to delve down into how the heart's organized as a biological structure. So we'll take this panel and we'll describe it as the four-box model of the four-chambered. You could read mammalian or even avian heart. I'm going to simplify this a lot, and we'll work on the anatomy and some other pieces. We're going to start here with this box. And I'm going to label this box as the right atrium. Now, it's worth taking a moment here to talk about the convention. Whenever we lay out a heart, whenever we draw a heart, and we talk about this as the right, the way we define it is as though we're looking down on another person's heart. So it's as though I'm standing here, and my chest is flayed open, and you're looking down on my heart. And that will be the right side and the left side. It's sort of like stage left and stage right. So what we're going to do here is, it's not as though this is the right side. This is the right side of the heart as you would be viewing it. So this is the right atrium. We're going to show it simply as a room. We're going to see that the entrance to the room here is through a structure called the vena cava. The vena cava is formed by the superior and inferior vena cava. The directionality of flow here is for the blood to come in this direction. And this is going to be oxygen poor blood. This is effectively oxygen which has gone, this is blood that has gone through the systemic vasculature, and most of the oxygen has been extracted from that blood, and the deoxygenated blood is coming back to the heart through this venous system. It's entering the heart through the vena cava. The atrium serves as a kind of receiving unit. This term atrium comes out of the Latin. And the atrium is this large room in say, a house, or more commonly these days, if you walk into a skyscraper. You walk into these gigantic buildings in a large city, and you enter this great big open space. That's the atrium. That's the receiving center, this receiving part room of that structure. Similarly here, this is the receiving structure. We're going to be receiving blood into this room, which is now the right atrium. The right atrium sits directly above this structure here, called the right ventricle. Now again, we're just talking about the chambers, we're just talking about the rooms. And so where this room is called the right atrium, this room is called the right ventricle, and there's a door between these rooms. This door has a name. It's called the right atrial ventricular valve. That's the nomenclature I'll normally use. I'll often refer to it as the right R, A, V valve. I find the nomenclature straightforward. It tells us where in the heart it is. It's on the right side. It tells us that it's a valve that separates the atrium from the ventricle. So it's the atrioventricular valve. So you can imagine here, this is a door between this chamber and this chamber, between the atrium and the ventricle. I'll refer to this normally as the right AV valve, but often in your reading you may find it termed something known as the tricuspid valve. This term tricuspid valve is pretty old. It comes from the early days of the Renaissance when they began doing gross anatomy. And when they opened up hearts of human beings and they looked at these valves between the right atrium and the right ventricle, they said there are 3 leaves to this valve, sort of organized in this, best I can do it this way. And then a third one coming out of the plane of the blackboard. So there were 3 leaves. You can imagine 3 separate doors in this doorway between the atrium and the ventricle. The flow now is through the door into the ventricular chamber. The only route out from the ventricular chamber here is into the pulmonary artery. It's worth taking a moment to describe what we mean by artery. An artery, by convention, carries blood away from the heart. And again, there's meaning in the terminology. It's the pulmonary artery. This is the tube that's carrying blood away from the heart toward the lungs. The blood that's going to come out of the right ventricle into this pulmonary artery is going to flow through the capillary beds of the lung. In the capillary beds of the lung, this deoxygenated blood, which is returned through the vena cava from the systemic vasculature, that blood is now going to receive oxygen in the capillary beds of the lung. So this blood is moving in this direction. Let me add this in here, we'll come back to it in a moment. There's now going to be another valve here, known as the pulmonary valve. In some quarters, you'll see the pulmonary valve referred to as the right semilunar valve. Again, I much prefer the terminology pulmonary valve. It tells you where that valve is. But this terminology of the right semilunar valve is left over from the Renaissance, and when they first started doing these dissections, they looked at these valves, they said it looks like a half a moon. So they're called semilunar valves. But again, pulmonary valve I think works better. So the movement through the chambers is from the right atrium to the right ventricle, out to the lungs. And then over here is another pair of rooms. This is the left atrium. The pathway into the left atrium is through this structure known as the pulmonary vein. Just as the vena cava was carrying blood toward the heart, here again the pulmonary vein is carrying blood toward the heart. Just happens that this pulmonary vein is carrying O2-rich blood. This is blood returning from the pulmonary capillary beds. So it's been oxygenated in those pulmonary capillary beds, and the flow now is to move through the pulmonary vein into the right atrium. Right beneath the room we call the atrium is this structure, which we name the left ventricle. Just as we saw on this side, there is an atrioventricular valve. So we can label this as the left atrioventricular valve. Just as the right atrioventricular valve had an alternate name, this too has an alternate name. It gets even more arcane. The left atrioventricular valve is said to be the bicuspid valve. Just as the term tricuspid said that there were 3 leaves, the bicuspid valve has 2 leaves. The other name for the bicuspid valve is the mitral valve. And again, this name nomenclature dates back to the 1400s. And when they first looked at this, and they looked at the mitral valve, they saw the 2 leaves, these 2 triangles. And they said, there are 2 triangles here. This looks like a bishop's hat. And a bishop's hat is called a mitre. And if you've ever seen the Pope in his full regalia, the Pope, because the Pope is the Bishop of Rome, the Pope wears these hats at these official functions. And they have 2 parts to them, one front and one's back. And they're these 2 leaves, effectively. And when the anatomists looked at this, they said, it looks like a bishop's hat to me. We're going to call this the mitral valve because this is, in fact, a bishop's mitre. Odd nomenclature. I much prefer left atrioventricular valve. It's just easier to hold on to. And then coming out of this chamber, again, this isn't anatomically correct, but coming out of this chamber is another tube called the aorta. And again, there's a valve or a door that leads out of this ventricular chamber. This is the aortic valve. Just as the pulmonary valve was said to be the right semilunar valve, this aortic valve can also be termed the left semilunar valve. But recognize that, in fact, it is the valve that lies as a doorway from the chamber of the left ventricle out into the tube of the aorta. And the blood flow here is going to be down across the left atrioventricular valve and then subsequently out into the aorta across the aortic valve. So that's the pathway through these chambers, the 4-chambered heart. The next thing I'm going to do here is I'm going to take this simple 4 rooms and talk a little bit more in terms of the biology and the structural biology of those 4 rooms. So any questions to there? Yes? So the blood from the aorta, the blood that goes out into the aorta goes through the rest of the systemic vasculature. So that blood is going to go out to your brain and your muscle and your liver. So all of the blood that's supplying oxygen to all of your tissues leaves the heart through that single tube called the aorta. But again, if you look at this above, recognize that those two pumps look just like the pumps we saw for the fish heart. It's just we have two pumps in series. One pump dedicated to pumping blood to the lungs, then once the blood comes back from the lungs, the other pump is dedicated to pumping that blood out to the systemic vasculature. If you spend a moment looking at that, you'll recognize that the volume of blood going out to the lungs has to equal the volume of blood going out to the systemic vasculature. Otherwise, one side or the other runs out of blood. And that'll be a concept we'll get to a little later. Anything else? So let's now take that image above and try to drill down a little farther and talk about the biological organization of the heart. And pretty much this is just going to be an elaboration of the 4 rooms. But now we're going to put walls on the rooms, and we're going to put linings on the walls. We're going to sort of paint or paper the walls. So if we start just as we did before on the right side, this structure is meant to represent the right atrium. Now again, this region in here, that's the chamber, that's the open room. This structure here is the atrial myocardium. The myocardium is the heart muscle. Now, the atrial myocardium is pretty thin. It's not generating a whole lot of force. The same pattern that we saw above occurs here. That's the right AV valve. And beneath the right AV valve is the open chamber of the right ventricle. But what we've added in here is this structure, which is the wall of the ventricle. The wall of the ventricle is almost entirely cardiac muscle, so we call this the myocardium. The myocardium is simply muscle, and coming out of this structure, I'm going to do it this way, is going to be the structure we described above, being the pulmonary artery. To make this consistent with the story above, there will be an inlet here, which is the vena cava. The vena cava is formed from the superior and inferior vena cava. So blood's flowing into the right atrium, flowing across the atrioventricular valve, and into the chamber, the right ventricular chamber. So these openings here that I have hashed in, those are simply open rooms, open chambers. This is a muscle wall. If we go to the other side, we see a pretty similar pattern. We see this thin-walled structure of the left atrium. This is meant to be the left atrium. This structure is intended to represent the left atrial myocardium. Again, we have this mitral valve, or the left atrioventricular valve, providing the only pathway into the chamber beneath. But that chamber now is surrounded by a fairly thick wall of muscle called the myocardium. Then in between the two chambers, there's a solid wall here. These two chambers are separate one from another, and this solid wall is known as the septum. And then just as we showed above, the output here is the aorta. And again, here's the aortic valve. Just as on the right side, there's a valve, a door that provides the door that allows egress from the chamber. This is now the pulmonary valve, also known as the right semilunar valve. Now, let me spend a moment here, because this question came up in the first period, which is let's look at the structure of this myocardium. The myocardium is organized into sarcomeres. Now, I'm simplifying this a whole lot because there'll be millions upon millions of cells, cardiac myocytes inside here. But recognize that what we have are typical sarcomeres, and that these sarcomeres obey the same rules as we've described for skeletal muscle. So that these are the thin filaments. And those thin filaments contain between them the myosin thick filaments. So this is intended to represent the myosin thick filaments. Now, everything we said the other day about skeletal muscle contraction pertains here as well. So that we'll get calcium released from the internal stores. The calcium will then bind to troponin C. That causes the tropomyosin to move. The myosin heads reach up and contact the actin-thin filaments. When the myosin heads reach up and contact the actin-thin filaments and undergo their power stroke, they shorten the distance between the Z lines. They cause sarcomere shortening. When the sarcomeres shorten, they're organized in such a way that what they're trying to do is they're trying to reduce the volume of this chamber. It's as though the walls are closing in. And they close in because of the contraction of these sarcomeres. But recognize that under normal circumstances, this thing is filled with blood. Because the chamber, the room, is filled with blood, the contraction now begins to generate force on that fluid. The fluid is non-compressible. So the contraction of the cardiomyocytes, the myocardium sarcomeres shorten. They generate more pressure on this fluid. One of the first things that happens when this pressure increases is the AV valve snapshot. These are pretty floppy valves. They're here basically allowing the blood to flow over those floppy valves, and then when the pressure increases, we snap that valve shut. Then the pressure increases and increases and increases until the pressure on this side is greater than the pressure on the far side of the aortic valve. This door swings open and the blood leaves. That's happening almost simultaneously with the right side. But again, the blood is flowing in across the AV valve. It's filling the room. It's filling the chamber. The chamber fills, and at some point then, the myocardium begins to contract. As the myocardium contracts, it makes an attempt to reduce the volume inside the chamber. But since it's a non-compressible fluid, the volume doesn't change, but the pressure goes up. And as the pressure goes up, it will first snap shut the right AV valve, and then as the pressure increases, the pulmonary valve will open, and that will allow the blood to squirt out into the pulmonary artery, carrying blood out to the pulmonary circuit. So the idea here is that these myocardia, whether it's the thin wall of the atrial myocardium or the thick wall of the ventricular myocardium, their function is to generate force. And they generate force, they apply a force to the non-compressible fluid of the blood. So any questions to there? Yes?
Can deoxygenated blood ever get into the right ventricle or the left ventricle?
Can we get deoxygenated blood leaking across to the other ventricle? Yes, there are birth defects. And I won't get too much into the details of this, but in utero, you don't have to worry about this side because you're not pumping any blood to your lungs. So basically there's a shunt between these two. Then at the time of birth, that shunt is meant to close off and separate the two chambers. In some cases, that doesn't close off properly, and then there's leakage from one chamber to the other. These days, we can go in and fix that pretty easily surgically. 50, 100 years ago, not so much. But now we can fix it surgically. So there are sometimes problems with it. Sometimes there are defects too in the septum. And there you get mixing between the left and right sides of the heart as well. And you don't, that's bad. So they'll then go in and sew those holes up as well. But again, and we can't get too much into this, but if you begin to look at the embryological origin of the heart, you begin to see why these defects occur. Because it starts out as a single tube. And even in fish, the single tube then forms an atrium and then a ventricle. Then to become a four-chambered heart, you get this complex folding system that then ends up with the right and left sides of the heart and those two pumps in series. The embryology is really exquisite. Yes?
Is the neuroactivity to activate the muscle fibers between involuntary and voluntary responses, is that purely differences where the neuron goes into the brain, or is it there's actually a process?
Okay, so you're bringing up a really, really fundamental point about the heart. And we're going to spend some time talking about this, but let me address it. In skeletal muscle, the lower motor neurons drive the muscle fibers. You cause contraction by action potentials in those lower motor neurons. It's effectively instructive. These guys aren't arranged quite that way. We're going to see that the contraction of the myocardium, particularly through a conductal system, has its own rate and rhythm. There'll be specialized pacemaker cells in the heart that generate the rate and rhythm of the contraction. Then in that case, what happens is the nervous system modulates that. So the autonomic nervous system, both the sympathetic and parasympathetic branch, is now going to modulate the rate and rhythm of the heart by modulating those pacemaker cells. And we'll spend some time talking about it, but it's good that you bring up this idea that we have two different mechanisms to regulate the contraction of the myocardium in the heart versus skeletal muscle. Skeletal muscle is always instructive, whereas the contraction of the cardiac myocytes is permissive because it's set up by the electrical system of the heart intrinsically and is then modified by the nervous system. Yes?
I think in later lectures we can go over how a pacemaker...
Later we're going to talk about pacemakers. We're in great detail. We're going to talk about channels and all that. Yes?
Could you go over the flow of blood?
The flow of blood? Okay. So up above we simply have the flow of blood through the chambers. The only thing we've added here is that we now have coming in through this tube the deoxygenated blood coming through the vena cava from the superior and inferior vena cava. Inferior vena cava is draining the lower part of the body. The inferior vena cava is mostly draining the head. The deoxygenated blood comes in here, flows across the atrioventricular valve and into this room, into this chamber. Then when the muscle on this chamber begins to contract, then that generates a force. That snaps this valve shut and squirts the blood out across the open pulmonary valve to the lung. Then that deoxygenated blood flowing to the lung flows through this complex capillary bed in the lung where it's oxygenated. Then oxygenated blood then is returned, and I should have drawn it up here, is returned to the left side of the heart through the pulmonary vein. Again, the nomenclature, it's returning blood to the heart. Blood is flowing toward the heart, so it's coming in through the vein. The oxygenated blood comes in this open pathway, in this open door, goes across the atrioventricular valve and into the ventricle. And the same story occurs. When the ventricular myocardium starts to contract, it increases the pressure on the blood inside the chamber, in the wall, in the room, snaps this valve shut. And then subsequently, as the pressure increases, opens this valve, and the blood goes out into the aorta. And what we'll do next time is we'll follow the linings of these cells. We're going to put an endothelium on the inside and a mesothelium on the outside. Yeah?
This is random, but